Um, we'll talk about this some more too. The engineered Colorado River is an extraordinarily young ecosystem as far as engineered ecosystems go. So, yeah, we will talk some about that today in terms of gene drives and quagga in particular. Um, the meeting information, I just remembered to start recording this meeting, so there will be a little blip in the front that's missing, but the meetings will be recorded, GoToMeeting, and then we'll put them, paste them on the website so you won't miss anything along with the presentations. Okay. So getting back to this. Before we go any further, I know this is kind of like herding cats, but if we could go around the virtual room and kind of make some quick introductions, I think it's always a good exercise. So if we could do that, if anybody wants to start. Anybody at all.
Scott Bryan with PAP.
White Hempstead City.
Andrew Truett, Bureau of Reclamation in the Lower Colorado Regional Office.
Brian Moorhead.
Esther Muniz. Oh, sorry. Esther Muniz from Imperial National Wildlife Refuge.
Dan Malloy, State University of New York.
Shane Maurer, Bureau of Reclamation, Upper Colorado Power Office.
Lance Corps of Engineers, Engineer Research and Development Center.
Prairie Dog, Colorado River Indian Tribes.
Great.
Ryan Fomuth, Aquatic Wildlife Specialist for Region 4 for Arizona Game and Fish Department.
Great. All right. Anybody else?
Hey, David, this is Barack. I'm not sure if my phone was muted, but Fish and Wildlife Service Regional Office, Region 2,
Good to have
Albuquerque.
you.
Good to be here.
Then Teresa Thome, I'm our aquatic ecologist at Lake Mead National Recreation Area.
Thank you, Teresa. All right, and I have to admit to a small vicarious thrill in having people try to do that. It's a little evil, but I can't help it. So as far as meetings go, yeah, we're doing some of these remote meetings, and like I said, I think they're a good idea. Our next one should probably be in person somewhere. I'm not quite sure where, but maybe we can have recommendations at the end of this meeting. Somewhere along the river, either Lake Havasu City, Page, Las Vegas, or Yuma. Okay, a little bit about direction. And like I said earlier, we discuss all aquatic invasive species because ecosystems don't exist that contain just one species. Right? There's always many involved, and that's what form ecosystems and assemblages. Right? And we do talk about some relatively technical aspects. I have to admit that I'm guilty of that. But we also talk about a lot of boots on the ground information, which is great, I think. And then information that, just from and to the general public. So like I said, the CRAISTF aims to be a clearinghouse of information regarding all aquatic invasive species in the Colorado River. So if I can try and get out of here. All right. And next, we're fortunate enough to have Teresa Thome from the National Park Service talking about AIS team efforts in Lake Mead National Recreation Area. Teresa, do you want me to just forward the slides for you, or is that easiest?
Yep, let's just go forward with that. That sounds great.
Okay. You are ready.
All right, so welcome everyone. I'm excited to have a chance to kind of give you a brief summary of some of our efforts here at Lake Mead that we've really focused on aquatic invasive species. And this work is not possible without the many partnerships that we have existing and hope to continue to have in the future, as well as new ones that we haven't even thought of yet. So again, it's not just the Park Service that's doing this. There's all kinds of partners that we are very fortunate to work with. We've done a fair amount of outreach and education since we have so many visitors coming to the park. We're using that as an opportunity to really be excited about what each individual can do about preventing aquatic invasive species from moving around. And then we have a fair amount of monitoring and research that we are doing as well. If you want to forward to the next slide. And if you wanted to see it in a kind of an infographic way, this is our newest kind of outreach tool of some of the just numbers of what we're trying to do, tied to talking with people, focused on programs, and then our partnerships. And last year in 2016, we had 7.4 million visitors to the park. In the year, well, last year for 2016, we sold over 36,000 vessel passes. So that's just when you launch a boat or are going to be using a boat within the park, we ask for people to purchase a vessel pass. That's not necessarily, day use pass. You can buy a pass for the year. You could buy a pass for a couple days. So, but that's the number of passes that were sold. The average number of boat trailers that we counted in our parking lots over a weekend, 4,451. So that just kind of gives you a scope of, of what we're talking about when we're saying we have boaters in our reservoirs on Lakes Mead and Mojave. So that's not just an average summer weekend, not necessarily a holiday weekend. And if we were going to wash each one of those boats, we would, assuming that each wash takes 20 minutes, which some are much longer than that, we'd need 61 days to wash all those boats. So anyway, that just sort of puts things into perspective. And then this last year in 2016, we educated over 52,000 boaters about aquatic invasive species. So that's not just our contacts on launch ramps and marinas, but some of our directed outreach materials to the boating community. And that's not just, hey, here's a pamphlet. Those are meaningful one-on-one interactions and really talking about aquatic invasive species. So, some of the other statistics and information you can sort of see on the slide, but there's a fair amount of work that we're doing, and we tried to kind of summarize this in a little different way. But again, our main message has been the clean, drain, and dry. Probably the next slide. And if anyone has any more questions on this, I'm happy to answer those. But we can, there you go. So just to give people kind of an overview, if you're not familiar with Lake Mead, we span 2 states, both Nevada and Arizona. We protect 1.5 million acres, and not just Colorado River, but that's the Mojave Desert as well. And we do have 2 reservoirs. Sometimes Lake Mojave gets ignored, but we don't. But we do have our 2 reservoirs, Lake Mead and Mojave. And again, last year we had 7.4 million visitors, a lot of trailer boats. And then just within the park, we have space for 3,972 boat slips. So that's just in our marinas currently active right now. We have that many spots for slipped and moored boats. And we're interested in that because when we've done risk assessment for boating, those slipped and moored boats, because they have the potential to be in the water for, more than 5 days, we see those as the highest risk for potentially transporting aquatic invasive species. So that's we've based on our risk assessments and the scientific panels and things, that's what we've really targeted our mandatory inspections. And outreach to is that those slipped in more boats, the people that have permits to have their boats within the park for, up to, who knows how long, but at least, more than 5 days. And then for the other boating community, the clean, drain, dry messaging, for day use boaters, that's really what we've focused on, that component of the required inspections and hot wash treatments, as well as our outreach. Next slide. Again, I mentioned the partnerships and how important this work is for us with the, not just the National Park Service, but we work very closely with both Nevada and Arizona. And then the Great Basin Institute helps us staff our launch ramps for the summer with our intern program. We are coordinating with other national park units, including Grand Canyon and Glen Canyon, as well as many other federal agencies, the Bureau of Reclamation, USGS. We really are trying to, be coordinated with all these other agencies, as well as our state and municipal and our university partners as well. Next slide. And so some of our outreach and education, here's one of our outreach teams. Some of our statistics here on the next slide. Sorry if there's a little delay. But since we really want to focus on the one-on-one contact, not just giving out pamphlets and brochures. So really since 2015, we've really focused on specific aquatic invasive species outreach messaging. And we've had different sizes of teams, but usually around 10 to 15 interns, and focused on our ramps during the busiest seasons, typically May through September, covering Memorial Day and Labor Day. And again, since they're really focused on the weekend contacts, the weekend boaters, that's we've focused on weekends for the busiest times. And just to see, the 34,000 visitor contacts, and again, that's not just, hey, here's some information. If you need a hot wash, this is where you go. It's, do you know about aquatic invasive species? Do you know what they are? Have you heard about quagga mussels? Do you know about, maybe you've heard something about the Great Lakes and having invasive species, and tell us what you know, and did you know that you can make a difference? So those types of interactions. Next slide. We've also done a fair amount of outreach and education activities off the launch ramps at, education-based programs at schools, in our library, at our visitor center, at special events and other types of activities outside the park, but also in the park too, but away from the launch ramps, just to try to reach, a different community, or maybe some boaters that haven't been to Lake Mead but are thinking about it. So we've really tried to go to these special events. In fact, this last weekend we were at the Get Outdoors Nevada Day, and we were stationed right by the Bass Pro Shops, where they were having folks practice being in a kayak, and we just had the safe boating messaging of wearing your life jacket, but also as appropriate for using a kayak or canoe or any type of watercraft, the AIS messaging as well. Next slide. And then, just also other activities. We're very focused on some of our scientific research, and also getting the message in a more of a professional science setting, being involved with our Lake Mead Science Symposium, which we have coming up again in February of 2018. And David also was part of this at the Association for the Sciences of Limnology and Oceanography at their national meeting when it was held in New Mexico. We were able to have a public session about the complexities of aquatic invasive species management in the Colorado River Basin. And so that was a really fantastic opportunity to have a special public session within a professional meeting. We're also involved with our interagency work group, the Western Regional Panel as well, and we're working on other publications tied to our research. Next slide. Yeah, so then our monitoring and research, we've been doing consistent monitoring, and then not just the National Park Service, but this ties in with all of our partners. Our interagency monitoring with the villagers, this is conducted with both the Southern Nevada Water Authority as well as the Bureau of Reclamation. And so the villager sampling has been ongoing since even before, well, since 2007, but prior to that work, it was really focused on monitoring the zooplankton and phytoplankton communities in both Lakes Mead and Mojave. And so this is ongoing work. We have our interagency monitoring action plan in place, looking at a whole variety of things, but that includes related to quagga mussels, looking at soft and hard substrate populations of quagga mussels, and that's been going on since 2009. And then we've done some targeted research on best management practices to look at, you know, how effective are the treatments that we're recommending right now. Next slide. And so more specifically with the best management practice research, we were looking at the feasibility or possibility of outboard engines transporting viable villagers, as well as looking at desiccation of adult quagga mussels. And that's tied directly to the 100th meridian recommended dry times for our region. So we were just testing that. For the outboard engine work, we first needed to develop a method that we could use to even just look at number of villagers in an outboard motor, and then test a different motor. So that's really what we did. And for the, again, for the quagga mussels, just looking at, we're trying to verify the different, or refine some of the recommended dry times for boats. Next slide. And so it's always good to have a few photos here, just so you can see what we were trying to do. We drove a transect. This is for the outboard work. We drove a transect on the boat to inoculate the engine. And next slide. These are going to be kind of fast. So we drove the boat up to our wash station. It's about a mile, not quite a mile, or I guess half a mile up to our wash station from the launch ramp after we drove a transect, and we did this with different size motors. Next slide. We're able to flush the engine into a bucket, and then you can look at the next slide. From that bucket, we were able to filter or put that into our a real fine mesh filter, and we were able then to look at, first of all, measure the volume of water that was in the engine, and then look at the number of villagers that were present in the motor. And we did different treatments, next slide, where we either did or did not do a hot water flush. So we had, different treatments of this. Next slide. We were able to test a 25 horsepower Mercury, 60 horsepower Yamaha, a 225 Yamaha, as well as a 250 horsepower Yamaha. So we tested the different outboard engines. We hope to continue to do this with different sizes and different types, but that's what we had available at the time. And just so you can see, I think if you click one more time, you'll get some red circles. There you go. And click one more time. All right, so just so you can see, we had the two different treatments. Whether we had the control, so you can tell that the control was just being able to look at the number of veligers within 1 liter of water that we scooped from the same area that we were doing our transect, and then we had our water that was coming out of the different engines without being treated, and then after an engine flush. So really the take-home message from this is that the only motor that we even saw any viable veligers was the 225 Yamaha. We had what we think was 1 veliger. And it also, the other thing to note here is that definitely after an engine flush, there is no veligers. No. So first of all, without a hot flush or a hot water flush, there's pretty low risk from having any veligers even or viable veligers in the water that's within the outboard motor. But then if you do an engine flush, I'd say you've eliminated the risk. Plus, also the volume of water that's held in these engines is also very small. So that's another thing that we noted, and that's on the next slide. You can see these engines are not, the outboard motors aren't really holding a lot of water. So that's the take home there. Next slide. All right, so moving on to some of our aquatic, our AIS with the quagga mussels, the adults. We were curious to see, just how long does it take for an adult quagga mussel to die when it's out of the water? And next slide. So we had our scuba team get some hard substrates that were covered in quagga mussels, and we hung them in some little mesh bags in our marina. So this is a covered marina. They're not exposed to direct sunlight, and we had different dry times from 0 hours all the way to 168 hours. Just looking at, after this length of time of these quagga mussels being exposed to the, regular conditions, how many of them were alive. Let's see, next slide. So we did this work in June, also September, March, and December. And so we conducted this work, basically we're trying to get the really kind of the different seasons that we have in Nevada. And here, June, you can see the average temperature, it's around 100 degrees. That's the orange line, the air temp. And for June, really we start seeing a lot of death just at 4 hours. And then at 6 hours, things are mostly dead. There's just a few kind of stragglers. And then after 12 hours, there was nothing alive. And that was similar in September, seeing death after 4 hours and, between 4 and 6 hours. And then again, after 12 hours of exposure, everything was dead. So that's encouraging because in June, our dry time for the area is 3 days. So that's very protective. Next slide. And then again in December and March, you can see a little lower temperature for this time of year, but there was some after 48 hours, there was still some things alive, but nothing after 72 hours. And depending on where you are, our dry time in this time of year is around 15 days. And so again, that's very protective when we're seeing complete death of all these mussels after 72 hours. And again, they're covered, they're not in direct sunlight. It's relatively high humidity during the times that we were testing. In fact, every time we've done this work, we've had rain. So that's interesting to note that even in, we think, kind of better conditions for trying to encourage them not to die, they were, after 72 hours, they're completely dead. Another thing that we looked at was if there's a difference with size, if the larger mussels were able to hold on longer, but we didn't see any significant difference between the different size classes. We measured all the mussels and just to see if there was any significance in how long it took them to die, whether they were small or big, but there wasn't anything. So next slide. So to summarize, we were able to come up with a reproducible method to look at our, veligers in outboard engines, and we hope to continue that, with different types of motors, and then also different boats. I know we focused on outboard engines right now, but we could try to do something with inboard engines and different types of boats as well. For the outboard engines, there's really just a very small amount of water that was present in the engine types. And again, with the messaging of the clean, drain, dry, I think that is also probably very effective. And then we just didn't see viable veligers in that raw water in the outboard engines. We think maybe just the action of them being sucked into a motor is enough to really break them apart, and that's what we saw. We saw little broken pieces of veligers. And for the adult quagga mussel desiccation, the quarantine timetables right now are very effective, if not overly protective. But for our area over the year and this, for the next iterations of this research on the next slide, really looking at this type of work, the desiccation experiments in other climates would probably be very beneficial. I know David Wong is hoping to do some additional work in areas like New York, Wisconsin, other states. And so that would be really informative. As well as for the veliger work, we'd like to try different manufacturers, types, and sizes. And again, maybe do a different type of study showing for sure life or death with the veligers with staining protocols that are out there. All right, so that next slide, that's all that I've got. Really just focused on, again, our work with our partners is very important to us. We are still hoping to do the best we can with outreach and education to the public, people visiting our park, as well as to different events, really just preaching the message about what each individual can do to prevent aquatic invasive species. And if you have any questions, that's my contact information, and please don't hesitate to reach out to me.
Thank you, Teresa. There was a question on chat that I can't seem to open.
Yes, I can't. I see, let's see. Was the time of the month considered when the veliger engine treatment was tested? Oh, okay. So this is a good question. And so actually, David, if you could click to more time to the end there, slide 27 at the very end slide, I have a slide about our veliger abundance. And so this, I mentioned before that we do our veliger monitoring, and this is pretty critical work to do, understanding your zooplankton and your phytoplankton community, because what we see, this is just looking at the abundance, but we also have samples throughout the water column looking at densities of the different of veligers, but also our other animals and other algae and phytoplankton in the water column at different depths. And for Lake Mead, we see the highest density of quagga mussel veligers at between 30 and 40 feet in Lake Mead. Other lake systems may not have this same density of veligers. And so if you're driving your boat on Lake Mead and you're pulling water from 30 to 40 feet down, you're probably having a tough time with your boat. I mean, people are boating at the surface of the water, and we when we did our control, we're only seeing, probably one, I think it was, one veliger per liter was our density at the surface. And that may change at different times of the year. We don't see that change in our seasonality in Lake Mead of veliger density. But again, a different water body may have veligers present in different locations in the water column. And so for those waterways, outboard engines may pose a greater risk. But again, we're looking at risk from our perspective with science completed at Lake Mead and Mojave. So, but that's a good question. And so we did look at time of the month, but again, we don't see a big difference throughout the year of seasonality of where veligers are present in the water column. Does that get at your question, Terry?
Yeah, thank you.
Any other questions for Teresa?
Teresa, Scott Bryan with the Central Arizona Project. You had just said in your answer to that question on that you're finding the majority of your veligers at 30 to 40 feet deep. Any thoughts on whether that's surface temperature related, if it's light penetration related, if it's a combination? Any thoughts on that?
Yeah, I mean, there could be some temperature, but that's really where we're seeing the greatest. I guess it's really the thermocline and light penetration.
So they're found at the thermocline?
Yeah, that's typically what we've seen. And now the other part of that is they're not all alive. There's some that are dead in that layer as well. But that's really the greatest density.
Okay, thanks.
Teresa, I had a question. I'm sorry. Go ahead, Brian.
Excuse me. Do you have an idea on the dissolved oxygen at this level, the 30 to 40 feet?
I could look that up. Right off the top of my head, I don't have that.
Okay. Thank you.
Teresa, do you see any relationship between types of phytoplankton and veliger abundance at all?
I am so glad you asked that. We, in looking at the data that's been collected with SNWA, and Todd Tejan has worked really hard on this as well. We haven't really seen any change in the phytoplankton, the zooplankton community, other than veligers are present. That's been the biggest change. I just see a note here from Todd. He sent in the chat that the DO is at least 5 milligrams per liter, probably 6 to 7 at 30 to 40 feet.
Gotcha. That's kind of what we see in lakes in Arizona in general, probably in the Southwest, is that there isn't much. Oh, I see. It's interesting, Todd. Thank you. We don't see much of a growing season between winter versus summer for phytoplankton. There's just climate is not a limiting factor necessarily for phytoplankton abundance. It just doesn't get that cold. So do you think that might be part of the reason why you don't necessarily see a huge relationship between villager abundance and phytoplankton density, maybe?
Yeah, or that it's a flowing river system that is now a reservoir. I mean, just the kind of that shift, and there's still I mean, who knows?
Right. All right. Any other
Plus,
questions?
it's a pretty oligotrophic system as well. So
You're right. Ooh, yeah, less than 5 micrograms per liter. That is.
and we've had a really, really difficult time too with doing any type of predictions using, modeling and the chlorophyll numbers. It's just, it's so low.
Yeah, and that's one of the things I was going to talk about today, but I think I'm going to save that for another presentation, is that modeling phytoplankton in lakes in the Southwest is really difficult, first of all, to find sometimes even any relationship between phytoplankton and nutrients is difficult. I mean, we do see that, but it's at a gross scale. It's not like what you would see in other places of the country. So, yeah, modeling and making those predictions of phytoplankton abundance, phytoplankton type is very difficult. All right. Any other questions?
All right. Well,
for Teresa?
No, I mean, just to say that we continue to do this work, and we're here, we're working hard. So thank you very much for the opportunity to present some of our work from this last year.
And thank you very much for doing so. Now, if I can, there's a big lag on my screen here, so I apologize for the delay. There we go. There we go. All right. One of the things I did want to talk about today, and like I said, I had some other things. That's a good point, Todd. One of the things I wanted to talk about today is what I've been talking about at other meetings, and that is, CRISPR, and the CRISPR-based potential for a CRISPR-based gene drive in the Colorado for quagga mussel, and for other species as well. But let's face it, we're generally talking about quagga mussel when we're talking about gene drives, and so that's one of the things I wanted to kind of go over today. And I wanted to talk a little bit about, I know I kind of go back on these presentations often and talk about, what CRISPR is and what gene editing actually is. And I'm never quite sure who's going to be at each presentation or meeting. So there has to be some logical starting point, and there might be some duplicability between this and other presentations, but it's probably worth it. Especially for people that this might be a new thing for. There are other type of gene editing programs, constructs besides CRISPR. Some have been around for a while. We've talked about zinc finger nucleases before, but there's also meganucleases. There's transcriptor activator-like effector nucleases, or TALENs. Meganucleases, they're generally engineered versions of naturally occurring restriction enzymes. Meganucleases has been kind of challenging, honestly, for most academic researchers, because the DNA recognition and cleavage functions are intertwined in a single domain. In other words, they're very large. By contrast, the DNA binding domains of ZFNs and TALENs are distinct. It's more straightforward to modify the DNA binding specificities of ZFNs and TALENs than it is of meganucleases. But all three of those have had their challenges, one of them being cost and time to actually do. And now we have CRISPR, clustered, regularly interspaced short palindromic repeats. And like I said, we've talked about this before. Cas9 or the CRISPR-Cas9 system is unique, and is probably one of the largest deals in science in quite a while. At least since PCR, polymerase chain reaction. And it's probably going to be increasing in scope and magnitude into the future in terms of what we can do. We have a genome core at the U of A, a genetic research core at the U of A, and pretty much in them, along with biomedicine, pretty much 24/7 working on CRISPR-related products now. So it's something that is going to be increasingly used in the future for all sorts of things. Okay. The nice thing about CRISPR is, compared to the other systems, that the chance of mutagenicity is lower. Uses guide RNA, works well. The cleavage points are cleaner. And it's really inexpensive and quick to do. That is the whole breaking point of breakthrough that's associated with CRISPR technology, is that it is relatively cheap and easy to do now. And the reason we use genomic editing is obviously for things like biomedicine, anywhere from cancer research to cataracts to diabetes. I mean, it's all out there. And these are things that people are looking at, like I said, constantly. And rightfully so. It's also used in agriculture, crops and livestock. Actually, it's not used yet, but the potential for its use is being looked at. Increasing yield, disease resistance, pest resistance. All right. It's also used in industrial biotechnology, biofuel production. It's currently against the law to use genome editing in any human embryo that will be allowed to develop beyond 14 days. Okay. It's possible to do it for longer, but it's against the law. And there's, we can talk about the ethics of that all day long, but we won't. And it's also used to create gene drives, okay, especially for things like invasive species, which is what we're going to talk about. And we'll get into this more in a little bit, but normal Mendelian inheritance, 50% chance of passing one gene on to subsequent generations. All right. With this mutagenic chain reaction that we now call gene drives, there's 100% chance of passing that same gene on. It's not always 100%, but it's usually pretty close to it. All right. So it's a form of biased inheritance. And we'll get into the mechanics of that in just a little bit, but just the fact that we can do that, there's nearly 100% chance of passing on a mutagenic trait onto future generations is a large deal in the world of science. And I'm sure everybody's heard about that, and will continue to be into the indeterminate future. And there's always risk and reward, associated with any new technology. And sometimes our technological capabilities far outpace our full understanding of what that technology means. I mean, we know quite a bit about how ecosystems work. We know quite a bit about the physiology of organisms. We know a lot about genetics. But you mix all those things together, and it becomes increasingly more difficult to figure things out. Right? And it is, due to lacks in our knowledge, our current state of knowledge, it's impossible to calculate all risk beforehand. There are going to be unforeseen circumstances of almost anything that we do. Right? The nice part about a risk assessment framework is we sit and think about those unforeseen circumstances as much as humanly possible beforehand. And that's about the only way that we can minimize risk as much as possible. Can we ever minimize all risk? Well, no. I was actually at a driver safety training class the other day. Yeah, don't ask. And one of the take-home messages was, you must eliminate all risk from operating a motor vehicle. And I don't know how to do that. I mean, just walking out to your car, you could get struck by lightning. So there's no way to minimize, to get rid of all risk. But it's inherent and incumbent upon us to try and minimize that as much as possible. So I sent out an email about this seminar earlier, and that seminar is based on this publication, and that publication is at those addresses right there. And it's also on the website. And if you haven't listened to the seminars, you probably should. They're really fact-filled. They're interesting. There's a world of knowledge in there about what we can do and perhaps what we should try and figure out before we actually do some things. All right, but that is the citation, and I will use excerpts from that citation throughout this presentation. A few questions that seminar raised, all right, is, will inheritance of the genetic construct remain constant through generations, right? I mean, that is one of the driving forces behind gene drives, is that we want that to remain as constant as possible to perform whatever function that we want it to, and to affect a population the way that we expect it to. Okay. It doesn't happen all the time. Like I said, it's 100% biased inheritance, but it's not quite 100%. We also, what is the risk to non-target species? I mean, we're changing the genomic construct of organisms, and we're not quite sure. I mean, we can kind of figure out how that would affect that one organism, but we're less sure about how that will affect other organisms. Are we changing it in a way where we'll have any type of interaction with a closely related organism or even a non-closely related organism? We don't know that yet. I think that's one of the things that we need to think about. Also, how reliable are specific molecular markers? In other words, we add things usually to gene drives so that the mutants are readily observable in the field. Whether it's a color, that we use for fruit flies and Drosophila. I mean, they've proven that they can do that. Could we do something with quagga? Probably. But that is one of the aspects of a risk assessment for gene drive, is being able to readily and easily as possible observe those things in the field. All right, so let's say something goes horribly wrong, unintended, potentially harmful effects. Is it possible to address those, unintended, potentially harmful effects? Yeah, we try. I mean, there is reversibility of some of these gene drives now. But then again, we're not even sure about the consequences of that reversibility. And one of the reasons I did want to talk about this today is because every time I talk about CRISPR or quagga-based gene drives, there's a lot of excitement, and rightfully so. And I'm not arguing it could be a very valuable tool. I'm also arguing that potential risk is quite large right now until we have a better understanding of things. And ecological risk assessment does a pretty good job of trying to address that unintended risk or unknown risk. Right? And it is a quantifiable thing. It's not, narrative. It is a suite of probabilistic decision-making tools to evaluate potential benefits and harms under conditions of uncertainty. Well, conditions of uncertainty is a pretty broad term. It's not that ecological risk assessment can, like I said earlier, evade all risk. It's not going to be possible. But it is a humanistic thing where we should, in ethical criteria, for us to try and go back and minimize that risk as much as possible before we implement anything as potentially powerful as a gene drive. And in their definition of risk, it's the probability of an effect on a specific endpoint or set of endpoints due to a specific stressor or set of stressors. Stressors don't usually exist in a vacuum. They just don't. I mean, unless it's a very large one, right? Like a volcano or asteroid or anything like that. Usually the things that drive evolution and the things that drive Mendelian inheritance and gene drives in general is that it's slow change over time. I mean, I'm a toxicologist, and I can tell you that I can expose an organism to a no observable effect level if it's just one toxicant and one effect in the lab, if it's just one toxicant. But we don't live in a world where there's just one stressor or one toxicant. I can also tell you that I can introduce something to a suite of toxicants or a suite of stressors, and there is an effect even at levels below what there is under a single stressor, single response dose. So stressors are additive. Okay? And their definition of a stressor is any agent with a potential to alter a component of the ecosystem. Well, that's just about everything. All right, so we're using pretty broad definitions, but kind of have to start somewhere. An effect is any potential beneficial and or harmful outcome. Endpoint, societal, human health, or environmental value that is to be managed or protected. And often that is defined by regulatory requirements. Okay, regulatory requirements and in law, I mean, it should be science-based in general, and it usually is. And sometimes it is more than other times. There's at least three major steps to environmental risk assessment. There's problem formulation, there's characterization of exposure, analysis, what they call it, characterization of exposure and ecological effects, there's risk characterization. And this is actually derived, a lot of it is from invasive species research. So risk assessment or ecological risk assessment is quantitative and deals with extensively with uncertainty. At least we try to assign numbers to things as best as we possibly can. It's also flexible. It's flexible enough to evaluate processes at large spatial and temporal scales. Yes and no. I mean, I'd have to say that what works in one area doesn't necessarily work in all areas. There's never going to be a magic bullet. There's never going to be one answer or that fits all. Okay, some of these things are going to have to be more ecosystem specific, and we'll talk about that in a little bit too. Just some case examples of this kind of ecological risk assessment framework. There's a lot of work been done on mosquitoes lately. Aedes aegypti and dengue fever, also things like malaria. So their definition of risk is the probability that gene-driven modified Aedes mosquitoes will decrease new dengue infections in children by 50%. The stressors, persistent of gene-modified mosquitoes in the environment. But the endpoint that they're trying to get to is a decrease in incidence of new cases of human dengue infections in children. Okay, it's really a good idea to have these things written out in a format and then actually go back and be able to quantify those things in between those individual boxes. And that's what ERA attempts to do. Probabilistic determination depends on probability, obviously. Cultural values and public engagement. I think public engagement should probably be a little higher. There is still kind of a creep factor involved with CRISPR Cas9 and gene drives in general, and I can't say that in some cases that's not warranted. Probably is. So public engagement and being open with them, and as far as the research behind what we're trying to do, here's everything that we could possibly have thought of to minimize the risk to the environment and to you. And then there's also always going to be some degree of uncertainty even after we've thought about things. There can be unintended circumstances. It's behooving upon us to have some protocol in place in case we actually see some of those unintended consequences. As far as regulatory oversight for gene drives, it's probably going to be a coordinated framework between the US EPA regulation of biotechnology, the FDA, and the Department of Ag, USDA Plant Pest Foley, APHIS, to assess those potential impacts of biotechnology. Those agencies under such a coordinated framework would have to abide by NEPA. Okay. And although NEPA does have many strengths, it's not a probabilistic assessment of potential risks. It's could be a potential major flaw. Some of the strengths and weaknesses of NEPA in general, it is a standardized approach. It's required by legislative law. It does support the collection of large amounts of information and data. And there are relatively clear reporting requirements. And there is public input. And the weaknesses is that it's not necessarily a decision science approach. And in all fairness, it was never intended to be that. Neither an environmental assessment nor environmental impact statement provides a quantifiable cause-effect model. Those are mostly narrative, even though there's certainly statistics and modeling done for those. It's not necessarily a cause-effect model. And analyses conducted as part of NEPA are not required to be probabilistic or report quantitatively on uncertainty. And that's where ecological risk assessment really has it all over, the NEPA process. Also, through some of these things, we use these processes, especially ecological risk assessment, to determine what our testable hypotheses will be to conduct further research. Something that NEPA necessarily doesn't do. They did have a rough framework for environmental risk assessment for gene drives. Part of the problem with this, and this is a great thing to think about, but we go from, phase 0, 1, 2, and straight, which is the development phase, straight into release. And it is really simplistic, right? Part of the difficulty comes in the planning phase because we have no real proof of concept. There has not been any gene drive in the United States in a large scale type of quantifiable experiment yet. And I know I work in a lab and in the field, and I can tell you that controlled laboratory studies often don't mimic risk in the environmental world. We try to mimic it. That's what we do, but controlled studies by their very definition try to minimize variability that does exist in the real world so that we can observe things that we can't with so much noise going on sometimes, right? They also try to think about alternative forms or methods of eradication or how to make a population less fit. And, in the case of quagga mussels, do any alternatives exist? I mean, besides containment, of course, and I fully understand how difficult containment can be sometimes. But we've had, Pseudomonas fluorescens, and we've tried red ear sunfish, and we've tried many other things. I don't know of any other tool as powerful as a gene drive that would truly knock back a population of quagga. Come on. Yeah, so what could go wrong? We still need more data in identifying and quantifying as much as possible all of that uncertainty. Sometimes we can think about things and they're difficult to quantify. Well, we should really attempt to do so. ERAs can inform the development of testable hypotheses in gene drive research, like I stated earlier. And ERAs are largely research driven. Okay. Yes, there is a large amount of public input, and there should be. And we do discuss things like cultural values and resources. But it could provide quantifiable endpoints to determine when to move from one phase to the other. That's what we're looking for. I mean, that box that I showed you earlier, of those 4 or 5 different boxes, was highly oversimplistic, and it takes a large amount of confidence in results to move from one box to the next. Okay. And like I said, I believe that quantification should be species, obviously specific, but also ecosystem specific, because ecosystems, it's the reason we have different species, it's because we have different ecosystems. So what works in one area of the world won't necessarily work in another. There were at least 3 very distinct features for gene drives that they list in that publication, and I like it. The first, obviously, is a gene drive is passed on from one generation to the next at a rate greater than that described by Mendelian inheritance. In other words, it has to be biased inheritance, and it has to be greater than 50%. Okay. A gene drive construct can have effects on other parts of the organism's genome beyond the target. It's a possibility. Right? I mean, we're editing genes. We're editing very small parts of genes, and we're really not sure of how that is going to affect, besides what we anticipate, what we're trying to do, how that's going to affect the organism as a whole. It could have other effects. And, you know, one of the things with ERAs in general is that gene drive modified organisms by definition are designed to spread along with their effects into a larger environment. It's an imperfect world. I mean, can they spread outside of that environment? It's a possibility. The proposed use of gene-driven modified organisms will be part of a system with a large amount of stressors. That's what I was talking about earlier, and multiple interactions affecting multiple species and a number of endpoints. So trying to quantify all those is extremely difficult, but we should try and do so. They are intended to spread, and you know, we don't even understand ecosystem science to the nth degree, even in a naturally occurring ecosystem. So in an engineered system, one that's highly affected by aquatic invasive species, it's even more difficult. When I've talked about trophic cascades in the past, you know, if we remove an organism, what wholesale, or at least reduce its fitness, you know, how is that going to affect the rest of the ecosystem? It's a really good question. You know, I think it's one that we could logistically sit back and try and think about and try and quantify. And I do think that multiple releases will definitely be needed. I mean, this isn't a case where, you know, you put one mutant male or one mutant female in a reservoir the size of Lake Mead, Mojave, Havasu, or anyplace else and expect it to spread to the rest of the population. And I'll talk about that in a little bit too, kind of numbers needed type of thing. There is, like I said, reversal drives, so you can effectively stop a gene drive in its tracks, and I'll talk about that in the next presentation. And like I said, you know, those are engineered as a fail-safe, but the impacts of that fail-safe may introduce their own problems that we're unaware of. We're not sure yet. So just to kind of reiterate, EPA's current framework and guidance documents under NEPA don't adequately address the assessment of multiple stressors and multiple endpoints. They were based on risk assessments for, like I said, single toxicants and a single response. And their effects are on a limited number of endpoints. And like I said, in all fairness, that's, you know, NEPA was never meant to do something like this. It was never meant to quantify things as complex as this. There's a lot of text in here. So Vanderbrink et al. Came up with some guidance about what an environmental risk assessment for gene drive should look like. And obviously, I agree. Build a digital map of the study site. In other words, identify the ecosystem in question. How far do you want this to spread? When do you want it to stop? Map out regions in the landscape that have similar land uses, stressors, and management goals. That's relatively easy stuff to do. Establish a priori the cultural values and protection goals that will determine the success of the assessment and decision-making process. Sure, obviously, that requires public input. And then we jump straight into this. Determine the interactions among the species and the ecological processes and functions that will be affected by the stressors. All right, the first three are not that difficult. That one will be very difficult. Determining those species interactions and ecological processes and functions that will be affected by the stressor that you've implemented in there, that is obviously the largest deal of risk assessment for a gene drive. It's also the hardest to get to. Okay. One of the ways we try to get to there is we construct that conceptual model that reflects the sources of stressors, habitats, all that kind of stuff. Okay. Then we use that conceptual model to organize all the information that will inform the cause-effect modeling. Okay. We'll get to the cause-effect modeling in a second. So here's a conceptual cause-effect model for gene drives, which I say some of this risk assessment is based off of invasive species research. So obviously, one of the first steps is to locate the source, the type of introduction. The stressor would be the species, the modified genetic element that you're inducing in that species. And we'd have to come up with some number that we would expect would carry this gene drive through that population. The habitats, in this particular case, would not be difficult to figure out. It's the effects. And again, we go back to the effects. That's the most difficult thing to try and quantify and to try and figure out. Not just the target effects, but the non-target and off-target effects. We can pretty much, come up with a relatively robust model of how to determine the target effects. It's far more difficult to determine the non-target and off-target effects, ecosystem-wide effects, and then the impact. Okay, do we see a reduction in population size in this particular case? Do we see a reduction of non-indigenous or invasive species? We can quantify that. And then we have all these confounding sources and stressors, which there's quite a few. And they just list a very few in there. Industrial pollution, nutrients, metals, pesticides, herbicides, oil grease, all that kind of stuff. And that list is far larger than what they have in that little lower left corner. Climate, inter and intraspecies interactions, phytoplankton, like we were talking about earlier, all sorts of things go into that little thing that they have there of confounding sources and stressors. But we need to think about those, right? And those should be part of the model. And one of the ways that we can try and determine some of these cause-effect models is with these Bayesian networks. It's a lot like recursive partitioning. I've used them before, and they work pretty well. But they're not perfect. It's somewhat of a neural network, but not quite. They do inherently incorporate cause-effect relationships and uncertainty that other models don't. So in that regard, they work pretty well. Right? It also generates multiple estimates of risk, which is good. So we have decisions. Right? And a lot of those decisions are going to be subjective, honestly. Do we do X if we can expect a result of Y? Sometimes not. So some of the recommendations that they came up with, environmental assessments and environmental impact statements required by NEPA are largely inappropriate tools to characterize the risks of gene-driven modified organisms. And I agree. They were never meant to be used for gene-driven modified organisms because they weren't around when it was developed. Ecological risk assessment would be beneficial to gene drive research. It can be used to estimate probability of immediate and long-term environmental and public health harms and benefits. And I say that rather simplistically, and I want to go back to how difficult sometimes that is to determine. It's very hard to determine what those causes and effects might be. they come up with a rather simplistic view of a Bayesian model for a cause and effect model, and I agree. I mean, that Bayesian networks work pretty well, but they don't work 100% of the time. And then one of their primary recommendations: researchers, regulators, and other decision makers should use ERA to estimate the probability of immediate and long-term environmental and public health effects. And I absolutely agree with that. I mean, researchers need to get together with risk assessment people and politicians and resource management agencies to try and figure out what we're trying to do. Another recommendation they recommend is to strengthen future ecological risk assessments for the use of field trials, and I agree. This should be any gene drive that is implemented or prone to be implemented should be field tested and verified. At the same time, sometimes field tests result in unintended consequences in their own right. I mean, I work in a biosafety level 2 secure facility, and I even know that even in those, there are some unintended consequences and escapes of certain things. So we have to make sure that whatever field trials we come up with is in a relatively safe and secure facility, where until we're ready to introduce them into the environment, they won't be introduced into the environment. That's a difficult thing to try and figure out too. Their third recommendation: to facilitate appropriate interpretation of the outcomes. Yes, there needs to be much more coordination between researchers and risk assessors. And something like this is to be planned for quagga mussel and the Colorado River. Well, the time to probably do that is now. Gene drives are moving at an incredibly fast pace. It doesn't mean we have to move at that same speed. We have the luxury of time to sit back and make these decisions based off of our best knowledge at the time. I have little doubt that a gene drive would be very effective for quagga mussel in the Colorado River Basin. What I'm less sure of are unintended consequences. Right? That's where that environmental risk assessment comes into play. And it is probably likely time to form a comprehensive group of folks, maybe here or elsewhere, that are both well-versed in risk assessment and gene drive research that's specific to the Colorado River Basin. That's all I have on that. Are there any questions? Did I confuse everybody? And for some reason, I'm not capable of opening chat. Hang on. I don't know why.
David, this is Barack. I got a quick question.
Sure.
Could you pose maybe to the group what would be an unintended impact from introducing the gene drive quagga into a system, and how we would go about, or I guess something that would trigger a reversal requirement?
Well, that's kind of in my next presentation, but it's a very good question. I think that the potential for harm, for bad things to happen, are lower in the Colorado River Basin than they are elsewhere, which I'll talk about in a little bit. But I mean, is there the possibility that it could, interbreed with another native species? I don't see it. I mean, I really just don't see it. Is there the possibility that you could create a mutant organism that actually is more fit than less fit? I don't see that either. I mean, but I do think that it's going to take more than my mind to sit back and think about it.
Sure.
I think it's going to take a lot of people from a lot of different perspectives to sit back and think about it, and then address each of those as they come up. Does that make sense? So in other words,
It does. Yeah, absolutely. I mean,
really,
I think about it and I'm just like, well, where would that go and what would that look like?
right.
What actually would trigger the need for a reversal in the system? I guess that comes down to the assessments that are required. So.
And I sit back and think about the same thing constantly, and I honestly haven't come up with one that's burning or at the forefront of my mind. Does that make sense?
Sure.
Yeah. There are many that may come up, and like I said, that's why they're unintended consequences.
Sure.
Right? But like I said, I think it takes a lot of people getting together, people who are more well versed in risk assessment than myself, people who are more well versed in the cultural aspects, to sit back and think about it. Is there any other questions? It is a lot to think about, I know. You're right, Terry. All right. Well, maybe this will help to answer some of the questions you raised, Brock. It's kind of this next presentation. Open up. So that was kind of the risk assessment for gene drives kind of generally. And like I said, I do think that gene drives should be one of those things that are engineered for the ecosystem in question. Gene drive that's going to work here is not going to work necessarily as well in the Great Lakes or any place else, okay? Because ecosystems are different. And I've talked about this in the past before too, and I think it's worth repeating probably over and over again, is that the Colorado River system, as far as an engineered system goes, is super young, 60, 70 years, which is nothing, ecologically speaking. It's a very new system. And there are things within that system that are still jockeying for position. I mean, when we built the Hoover Dam, did we ever expect that quagga mussel were going to invade? And were you talking about gene drives today? No, of course not. So yeah, it's a super young system. Things are still jockeying for position, trying to calculate and quantify and figure out what that biotic assemblage will look like in the future is very difficult to do. Oops. And I've talked about this before too, is that those founding effects and bottlenecks, they don't stop AIS that have evolved under strong selection pressures. And we've talked about environments just like that, whether it's Asian carp or whether it's quagga mussel. Those have evolved, those are the type of organisms that have evolved under extraordinarily strong selection pressures. And it is true, I mean, nature abhors a vacuum. So are there resources that are unused in the environment? I wouldn't say that they're unused. I'd say that there is room for invasive species to come in and utilize those resources, and obviously there is in the Colorado River. That's why we have quagga mussel. So, the Colorado River is extremely vulnerable to aquatic invasive species. Like I just said, and that's especially true under changing climatic conditions. We've had warming over the basin since we built those dams upon it. So there's another confounding factor. Current genetic variability of quagga in the basin. Well, we've talked about those. We've had many great presentations on those, from Sherry Pucciarelli to Alicia Jeanette to some other people. And overall, genetic variability is still relatively low compared to other areas in the world, especially Great Lakes and in their native range, sniper drainage. Heterozygosity will likely slowly increase over time. Even small amounts of genetic variability are observed in those areas away from the core founding population of Lake Mead. They're small, but they do exist. Right? So as quagga move into other areas, heterozygosity will likely increase too. And yeah, containment and the work that Teresa's doing and everybody else is doing is so vitally important. And we need to know those things. We need to have containment. We don't want, obviously, quagga to invade other places for economic reasons, but we certainly don't want them to invade other places for genetic reasons as well. We don't want to increase heterozygosity. Right? Statistics are not kind to even the best of containment efforts, and that's true. I mean, containment will carry us so far, I think. In some circumstances, it may prolong an invasion. It may even outright prevent it in some cases. Unfortunately, it is one of those things where, with something the size of a trochophore, with a species that's as fecund as quagga mussel, it's very difficult to contain everything. And we are constantly having to work against the ecological grain of natural selection in that containment. Right? Species want to spread. They want to emigrate from one place to the next. Nothing does that better than quagga mussel. Spatial variability in gene drives and risk assessment. Like I said, what's idealized for one area may not work in others. Ecosystems all contain very unique sets of variables, intraspecific variability. And gene drives and risk assessment should be done on a watershed or smaller scale. And they just should very much be ecosystem specific. These are really gross categories of lethal versus non-lethal gene drives. Non-lethal in this context doesn't mean a drive wouldn't be capable of eradicating a species. It means that the target genome, or the gene that you're looking at, doesn't involve reproduction or the lack thereof so much. You can target other things, other life history aspects. In the case of quagga, we could look at things like byssal threads, calcium uptake, calmodulin gene, other things could be targeted as well. And the result would be eventually eradication or making a population less fit. There are some pros with that. It's relatively low risk, low potential of spread, lower relatively potential of spread to other areas or species. It's less likely to be stopped due to genetic variability. One of the things about organisms in general is that fertility and fecundity is usually one of the last things to go. Species want to persist. That's a very well inherited trait. Right? So trying to push back against that can be done. Sometimes gene drives can be stopped just due to natural genetic variability that exists in the field. Such a drive would be labor intensive, logistically difficult to introduce. I'm not, all of them are going to be logistically difficult to introduce. Reintroduction would definitely be required, and it would require something like genetic swamping. And I'll talk about numbers here in just a few. I've talked about this in the past. Potential non-lethal genes to target. Quagga have an absolute requirement for dissolved calcium, and they use this calmodulin gene in order to acquire that calcium. And calcium requirement increases post-trochophore. Okay, obviously, because that's where we're using calcium for shell formation and everything else. Calmodulin, we have calmodulin. Everything has calmodulin. It's very important in shelled organisms, bivalves and mollusks for shell development. There's calmodulin-like proteins as well. Both CAM and calmodulin-like proteins are vital to shell regeneration and formation in all mollusks. Calcium-bound calmodulin forms a critical subunit for regulatory enzymes, phosphorylase kinase. Is it possible to target calmodulin gene? Absolutely. Absolutely. So that would be just an example of a non-lethal attribute that you could use in a gene drive. And then, of course, there are lethal or sex-based gene drives. Would directly affect reproductive potential in subsequent generations. Still requires a large number of FOT organisms to be introduced in the environment. Both of these will. The pros is that directly reduces population numbers through infertility. The cons, like I was just saying, reproduction is one of the most highly conserved traits of organisms. It's highly susceptible to genetic variability within a population, and this still requires a degree of genetic swamping. Right, and there's this. Gene drives thwarted by emergence of resistant organisms. That's just, and those resistant organisms are only resistant because of natural genetic variability that were in the mosquito population of which they were talking about. And there's also why fake islands might be a real boon for science. I don't know what that is about at all. So real quickly, we've talking about how transgenes or mutant alleles carried on via gene drive. How do we ensure the spread of an engineered genetic material through a population? And how do we drive a trait from heterozygosity to homozygosity? It's possible. There's in Gantz and Beer explained it quite eloquently in a paper in 2015, mutagenic chain reaction, MCR, which is basically synonymous with the term gene drive. That's kind of where the term gene drive started, is a mutagenic chain reaction. Loss of function mutations may only produce a mutant phenotype when both copies of the gene are mutated. In other words, there's your 100% being passed on to offspring. So yeah, eventually MCR in gene drives cause homozygosity of a trait. Yeah, we won't go through all of this, but basically it's a homology-directed response, or repeat, and just some of the genetics of gene drive are on the left. Mendelian inheritance, that unique trait, usually gets wiped out over time, more often than it's carried on. But in gene drives and that mutagenic chain reaction, maybe not in the F1, F2, or F3, but eventually it's going to be 100% homozygous for that trait. Okay, and when it is, populations usually go extinct. The speed and kind of the numbers that would be required to kind of initiate a gene drive depends on the effect of individual's fitness, on the rate of allele conversion, population structure, that kind of stuff. However, only 30% reproduction is required for a gene drive to progress, which is relatively low. If we could stop reproduction of quagga to 30% out there, which is the overall goal, that would definitely reduce its fitness. Just to kind of get some sense of behavior of this model, even a 25% reduction in fitness in this one results in a conversion efficiency of about 80%. I mean, even if you reduce, 0.01, if you introduced 1,000 mutant quagga for every million that happen to be in a lake, eventually, over 10 or 15, 10 to 12 maybe generations, that would become a homozygous population for that allele, which means it would probably not be there anymore. Okay. Go. Containment is always something to think about. Current boots on the ground containment efforts in the basin and between and amongst states is vital, and it would be if we introduced a gene drive or not. And we talked about having a reversible gene drive. And that reversibility should be incorporated as a countermeasure in any planned gene drive, whether it's for quagga or anything else. And they're not difficult to do. I mean, if we had some unintended circumstances, Barack, like you were talking about earlier, I don't know what those would be, you can introduce other organisms, right, to reverse that. You can reverse or stop a gene drive, and it's not difficult to do. You basically create an error in the lab at those non-homologous endpoints. And you create a gene drive resistant allele, and you introduce those into the environment. And they work pretty well. It would stop it. Fecundity against quagga mussel. Quagga mussel are they're extremely fecund animal. 1 million eggs per year produced by one female quagga. And due to generally warmer water temperatures in the basin than other areas, fecundity may be greater here than most other places. So that fact may mean that a gene drive based on fertility or fecundity alone could be stopped by even small amounts of heterozygosity in a wild population. That's true. But we also want a highly fecund species because that'll drive either a lethal or non-lethal gene drive deeper into itself, deeper into its own gene drive. Okay. Gene drives don't work on very long-lived, slowly reproducing things. They work really good on highly fecund, highly fertile organisms that have a shorter reproductive time. Aridity as a gene drive safeguard in the basin. Colorado River drainage area is 243, basically 1,000 square miles, but we're not concerned with all of it. I mean, we are concerned with all of it, don't get me wrong, in terms of quagga mussel. But, basically from Lake Powell down to, Cibola, something like that, which is a much smaller area to concern ourselves about. Yes, there are some off-target sites where we found quagga, including in Arizona, including in California. Not dismissing those at all, but it's just a much smaller area than some other parts of the world, with these sharp clines between the aquatic environment and the arid environment. And Teresa just gave an excellent presentation on desiccation rates of quagga. Right? There's a reason we don't see quagga walking across the Mojave Desert. I mean, they're an aquatic species only in boats. Unintended transfer would be their main mechanism of movement. And as difficult as containment currently is, and I understand that is completely, it would likely be far more difficult in a more mesic environment. So that is one of the things that the Colorado River Basin has going for it that other places don't. Their potential for interbreeding with other species, I'd say it's really low, man. I'd say it's really low. The most closely related species to quagga mussel in the world are zebra mussel, and they don't produce viable offspring. The most closely related native species, or species native to the basin, would be the California floater, and they're not taxonomically all that close. It would still be a good idea, and to do the due diligence. We should still look at the gene drive effect on any native organism. The most closely related organism native to the basin would be the California floater. As far as breeding with other species, it's fairly low. Something that's more difficult to predict are ecosystem or trophic level effects. And, I've talked about trophic cascades in the past, and they are difficult to predict. We see them in limnology all the time, whether it's Lake Tahoe or here or other places. We do know that there's microcystis outbreaks in the Colorado River, and we can't attribute those directly to quagga mussel, but they certainly can't be helping. I mean, that is a known occurrence in other places that quagga mussel have infested, as we do have large blooms of microcystis, which in turn affects things like zooplankton and other types of phytoplankton, even though these are fairly oligotrophic systems, like Teresa and Todd were alluding to. They are. We have relatively oligotrophic systems, but still a large amount of cyanobacteria. Like I said, there's no way we can directly attribute these microcystis blooms to quagga mussel.
Well, especially since we have microcystis outbreaks in places that don't have dracinid mussels.
Absolutely we do. Absolutely we do. And I see them in other places in Arizona and California and other places. We absolutely do. A lot of those places are not oligotrophic, though. I will say that. Some of them are. I mean, sometimes we do see a high chlorophyll, low nutrient type of situation. Right. So I'm not going to go so far as to say quagga are causing microcystis blooms. I will say that they're not helping it. I mean, they do have a selective they are selective for other species besides cyanobacteria. Okay. So even microcystis aside, would getting rid of quagga, would that change the way phytoplankton dynamics are in those reservoirs currently? Probably. Would it change? I mean, you can't have a benthic feeder that just feeds that much with the conversion rate despite that quagga has and not have an effect. I mean, there's just no way around it. And if you get rid of them, will there be an effect as well? Of course. Yeah, there will be. Trying to figure out that effect is difficult. There are many reasons why gene drive in the Colorado Basin should result in a large scale, at least a decrease of fitness in the quagga mussel population. And eradication should always be an ultimate goal. Like get containment. Containment's difficult. And we've done a really good job of doing that. Right? And we should continue to do so. Any gene drive and risk assessment should be designed for the ecosystem in question. I can't harp on that enough. There will never be a one-size-fits-all approach regarding gene drives. And going back to my earlier point of, coordination between those well-versed in the science of risk assessment and those researching effects, positive and negative of gene drive research is essential, and it should be early. It should probably start now. It should be the first step. Okay. And there should be a group likely coordinated specifically for the Colorado River of those people that are well-versed in the Colorado River. That's it. Does anybody have any questions? Did I totally confuse everybody? And I can't open the chat box for some reason. Hang on, let me try.
It's Barack. I just wrote in it. Thank you, David.
All right. Thank you, Barack. Anybody have any questions at all? I think this is something that deserves further discussion, maybe at some point in the future. We have a lot of other things in the AAS task force to discuss, and we'll continue to do that. But with the changing science and the changing tools at our disposal, changing basically every day. I mean, everything I talked about in that presentation was from 2014 on, pretty much. Some of it within just a few months. So we probably will talk about that more in the future, but also other organisms as well and other topics. All right. If there's no questions, then we will conclude. Okay, we're a little bit early. I usually like to leave 15 minutes at the end for discussion, but we will have one. Like I said, if anybody has any questions, you can either email Teresa or myself about her presentation or about mine. Thank you, Teresa. That was a great presentation.
Thank you for letting me present.
Absolutely, anytime. All right, we will conclude. Thank you all for being here, and we're due for another probably in-person meeting. This is October, November, probably sometime after the holidays, January or February, and I'll talk to people and we'll discuss about where to have that one. Being remote is nice, but there's nothing like being in person as well. So hopefully we'll have one of those soon. Okay.
Thanks, David.
Thank you much.
I'll talk to you soon.